Good evening, all of you. Good evening, students. So, how are you all? I hope you all must be doing great. So, just if my voice is clear, just let me know. Yes. Good evening, Sai. Good evening. Fine. So, I hope that the voice is clear. Great. So, now let's start with the topic. So, here today we are going to finish the chapter cell. So basically it is the last class for this chapter cell. So in this class, of course, we will conclude the chapter, and in the end we will solve some question directly which is asked in previous year NEET, so that you get the idea that exactly how the question is asked in NEET, and whether after covering these basic points can you solve those questions or not, right? So that you get a flavor of how NEET ask question. So that we will do in the end part. So let's first complete the chapter. So in last class, if you all can recall, so we were talking about the nucleus. So we have just started with the nucleus, in which we have seen this is the nucleus. Nucleus has these parts, in which we have seen that nucleus has nuclear membrane, or you say nuclear envelope basically. So this is the nuclear envelope. Now if we see, this nuclear envelope is double layered. So it has a outer layer and it also has inner layer, right? So nuclear envelope has an outer membrane and has an inner membrane. Then another part in the membrane is, see here, it is the nuclear envelope. Then in the nuclear envelope, small pores are present, which is called nuclear pore. Inside is the nucleoplasm, and nucleoplasm has a spherical structure called nucleolus, and the thin thread-like structure is present, which is chromatin. So this is overall the part of nucleus. This we have done in the last class. So part of the nucleus we have done. Now today we will see all these parts of nucleus one by one. So let's first start with the nuclear envelope. So the nuclear envelope, if I draw the diagram here, so nuclear envelope has a outer and a inner membrane present. So here let's see the diagram. Here we see what? There is a outer membrane, there is inner membrane. See how I have drawn this diagram. You can clearly see that this is the nuclear envelope. It has outer membrane, and it has inner membrane. In between outer and inner membrane is a space which is called perinuclear space. Perinuclear space. So this is part which I think is very much clear. So can we say nucleus is a double membrane structure? True or false? Nucleus is a double membrane structure. True or false? It is correct. Nucleus is a double membrane structure. Now, on the surface of nucleus, On the surface of nucleus, outer membrane, what is present? Tell me. On the surface of nucleus, outer membrane, this spherical structure I am drawing, tell me what is it? Quickly tell me what is it? The outer nuclear membrane have. Tell me what is this structure? Very good. Very good. Those who are asking, true. Very good. Okay. Shakuntala is telling it is ribosome. Very good. Sai is telling it is ribosome. Chandrika, ribosome. Very good. Venkata, ribosome. Yes. So we have seen in ribosome that ribosome can be present on the surface of ER also, and also can be present on the outer membrane of nucleus, right? So outer, it will be present all throughout, right? Also we have seen that the outer membrane of nucleus is continuous with which cell organelle? Tell me, again answer me. Name the cell organelle which is continuous with the outer nuclear membrane. Let me see who answer it first. Cell membrane, which is cell organelle which is continuous with the outer membrane of nucleus. Let me see who can answer. All those who are here, just only write the answer. No other thing in the chat box. The outer membrane is continuous with which cell organelle? Yes, Ritika has said first correct answer. Ritika has given the first correct answer. It is continuous with endoplasmic reticulum. See, this ribosome is present on the surface, but continuous means what? Let's say this is the outer membrane, right? And endoplasmic reticulum is present like this. So what we will say, right? So this is nuclear membrane, so and this is the endoplasmic reticulum. So we will say that the outer membrane of nucleus is continuous with which cell organelle? Endoplasmic reticulum, right? Very good. So many of you have answered ER, Kalyani, Vasu, Manoj, right? Lakshmi. So it is correct. Those who have answered ER, it is correct. Understood this point? Now let's move ahead. So we are not going to get into detail of ER, we have already covered it. Just remember that the outer membrane of nucleus is continuous with ER, and outer membrane also have ribosome present on its surface. Another question, who will tell which ribosome is present? This ribosome is 70S or 80S ribosome? I am waiting. Who will answer first? Let me see. Who will answer first? This ribosome present here, is it 70S or is it 80S? Tell me the answer. We have already discussed this also. Tell me, is it 70S or 80S? Mallikarjun is telling 80S. Very good, Mallikarjun. Reddy, 80S. Fine. But many of you are answering 70S. Remember, any ribosome present on the surface will always be 80S. Whether it is present on endoplasmic reticulum, that is RER, or present on the surface of nucleus. In both places, the ribosome will be 80S, right? So it will be 80S ribosome. Remember this. All of you note this down, that the ribosome present on the surface of nucleus will be 80S ribosome. 80S means it will have two subunits, 60S plus 40S. Two subunits are present. So this part is clear. Now, this nuclear envelope is not a continuous structure. Nuclear envelope is not a continuous structure. Actually, the outer and inner membrane fuse together and it form a structure called nuclear pore. Nuclear pore. So let's see a bit about nuclear pore. What is a nuclear pore? So nuclear pore basically nuclear pore actually, can we say that nuclear pore connect nucleoplasm with cytoplasm, right? So first thing, nuclear pore, it connects nucleoplasm with cytoplasm. See, this is the nucleus. Inside is what? This side is nucleoplasm. And this side is what? Cytoplasm. So isn't nuclear pore connecting the nucleoplasm with cytoplasm? Right? So nuclear pore connect nucleoplasm with cytoplasm. How a nuclear pore is formed? Nuclear pore is formed when the outer and inner membrane fuse together. Is this point clear? So this is all about the nuclear envelope. Remember the role of nuclear pore. Once in 2023 NEET question was asked from nuclear pore, right? So nuclear pore is also important and envelope is important, and have the reference of ribosome also. So we have seen the nuclear envelope part. Now we will go inside the nucleus. So inside the nucleus is nucleoplasm. Now this nucleoplasm, what we have seen earlier, that the nucleoplasm is divided into nucleolus and chromatin. So now nucleoplasm will be divided into nucleolus and chromatin. Nucleolus and chromatin. So we will see them one by one. So what is nucleolus? So nucleolus is this. It is a dense structure present inside the nucleus. So some characteristic about this nucleolus, let's see. First is nucleolus is present in prokaryote or eukaryote. So first thing, it is present only in eukaryote. It is absent in prokaryote. First thing. Second, where is nucleolus present? So it is present inside the nucleus. Then question comes, is it bound by membrane? It appears as a sphere, but does it has an outer membrane around it? So it is non-membranous. Means there is no membrane surrounding the nucleolus. Nucleolus is without membrane. Then the question comes, why it appear a distinct structure? Because it is a dense structure. Dense structure, and it is densely stained also. It is densely stained. Then question come into your mind, what is the role? What it is doing? So nucleolus is performing very important function. Nucleolus help in synthesis of or formation of, you can say it helps in formation of ribosome. Ribosome. Now, ribosome, earlier we have studied, ribosome is made up of two parts. Ribosome is made up of two parts. It is made up of rRNA and it is made up of protein. So the rRNA part of ribosome is made by nucleolus, and that is why nucleolus is also known as, it is also known as ribosome factory. It is also known as ribosome factory. Remember this name. This nucleolus is also called ribosome factory. Is this clear? Question if I ask. Now, so there are two factory we have studied. One was protein factory. What is protein factory called? What is protein factory called? Second thing we have seen, ribosome factory. So who is the ribosome factory? Nucleolus is the ribosome factory. But what is the protein factory? Protein factory. Protein factory is, protein factory, yes, Chandrika, again you are right. Chandrika, very good. Protein factory is ribosome. Last class we have discussed that. Ribosome factory is nucleolus. Note it down. I will write in this corner. See, protein factory. Protein factory is ribosome. And ribosome factory is? Ribosome factory is nucleolus. Understood this point? So we have seen the structure and function of nucleus. Now question can come, one cell will have only one nucleolus or it can have more than one nucleolus? So one cell can have one or more than one. More than one nucleolus can be present. Means here although it is appearing only one, but in some cases when the cell is very active, then more than one nucleolus can also be found. More active the cell, more nucleolus can be found. Yes, Mokshit, correct. Mokshit is telling that yes, it can be more than one is also possible. So correct. So here it can be one or more than one. Remember this. If statement is there that nucleus can have more than one nucleolus, true or false? So it is true. So function is clear, structure is clear, everything is clear. So now we will go to the next part of nucleolus. So nucleolus, once again I will summarize here because we are writing here. So summarize. Nucleolus, membraneless. Membraneless. It is dense structure, right? Then it is called ribosome factory, right? It forms what kind of RNA? rRNA. rRNA means R here stand for ribosomal. Ribosomal RNAs. Later on we will study that RNA actually is of three types. RNA can be rRNA, tRNA and mRNA. Not now we will study this, you will study in the second year. Second year you will study rRNA means ribosomal RNA, tRNA means transfer RNA, mRNA means messenger RNA. So we will see it in the second year. So this point you understood about nucleolus. Now next we will see the chromatin. So let's see how chromatin is present inside. So see here you can see clearly thread-like structure present around. So this thread-like structure will be called as chromatin, right? So let's see what is a chromatin. So let's give the heading chromatin. Chromatin. So first is they are thread-like structure. Thread-like structure. Very good. So they are thread-like structure, but what is its character? So if we study the character basically, and this is very key term, chromatin are nucleophilic. protein. Chromatins are nucleoprotein. Nucleo means they have nucleic acid. Nucleic acid. And they are also having protein. So what kind of nucleic acid is present? So it has DNA. It has RNA. Now what kind of protein is present? Histone protein and non-histone protein. Now in NEET previous year question, there was one question that which of the following is a part of chromatin. So is DNA a part of chromatin? Yes. Is RNA a part of chromatin? Yes. Is histone a part of chromatin? Yes. And is non-histone protein a part of? So yes. So chromatin will consist of DNA, RNA, histone and non-histone protein. This point is 100% clear to all. So this is a chromatin which form a thread-like structure. Now what is this chromatin made up of? So this chromatin actually is made up of, here as I said, DNA is the major part of the chromatin. So chromatin has DNA, right? Chromatin has DNA. Now this DNA is a thread-like structure. So let's first understand this point, that the DNA is a thread-like structure, right? Now this DNA will condense and form chromatin. And this chromatin will condense and form chromosome. Understood? In normal condition, the cell doesn't, the nucleus do not have simple DNA and do not have chromosome. They are present in the form of chromatin. So what we will say, a non-dividing cell. Non-dividing cell. What is present? Chromatin is present. But when the cell become dividing cell, what will happen? Here the chromatin, which is a thread-like structure, will get converted into chromosome. Now let's imagine this with one example. Let's say you have a long thread present, right? At our home, threads are present. So how we keep the thread? So we take this and we just rotate the thread so that they are not intermingled, right? So chromosome is also like the same. When we keep the thread and bind it and form a particular shape, then it becomes chromosome. If you unwind the thread and randomly open it, what is it? It is chromatin. So when the cell is not dividing, it is present in the form of chromatin. But when the cell is dividing, then it will arrange itself in the form of chromosome. So chromatin will appear just random, anything you draw, thread-like structure, random, but chromosome will have a particular shape, right? It will have a particular shape. So I hope this point is also clear, that inside the nucleus, chromatin and chromosome, how they are interchanged. In which case chromosome is present, in which case chromatin is present. Ultimately both are made up of DNA only, because in the chromatin also DNA is present, in the chromosome also DNA is present. So now if we see DNA, so now we will talk about the DNA. So structure of DNA, all of you, structure of chromosome, all of you must have studied about the chromosome till now, right? So let's see, first let's understand this structure, and basically both are the structure of chromosome, but both are showing some different points. So here if I see this structure, let's say this, so this is also one chromosome, This is how many chromosome? Tell me. This is also one chromosome. And this is also one chromosome. Then what is the difference? Both are one chromosome. Then what is the difference between them? So here, see, here one chromosome is having, this single is called chromatid. What is it called? Chromatid. And this one is also called chromatid. Now, so now what is the difference? Here this chromosome has two chromatid, and here this chromosome has only one chromatid. So chromosome can have one chromatid or two chromatid. The two chromatid present on the chromosome is called, they are called sister chromatids. They are called sister chromatids, right? So two chromatid together are called the sister chromatid. So understood this point? Now when DNA replication take place, then this will become after DNA replication. After DNA replication, a chromosome with one chromatid will become chromosome with two chromatid. It is just like you have a page, you put it into the printer and you get one more copy of it, right? It's just like you get a photocopy of this. So this is one, you get exactly same copy, so it form a sister chromatid which is exactly the same. This point will be very, very useful in understanding different concepts in the cell cycle. Chromosome with one chromatid, chromosome with two chromatid. Now other part, if you see at the center of the chromosome, at the center of the chromosome is the centromere. So another part of chromosome is centromere. Centromere, this also has one centromere, this also has one centromere. Then this is called the arm of chromosome. This is also called the arm of chromosome. So this is the arm. So this chromosome has how many arms? So you will say two arms, 1, 2. But this has how many arms? It has 4 arms, 1, 2, 3, 4. So arm of chromosome is also clear. So now let's see this structure. So here you can see this is the representing the DNA. The DNA coiled and it ultimately formed the chromosome. It is showing that coiling of DNA form the chromosome. Fine. This is the centromere, right? Then these are the arm of chromosome. See, there is a short arm, there is a long arm. Always short arm, long, many times the arm length is equal also. Then there is no short and no long arm. You can keep P and Q value will be the same. But in general, when the length is different, then the short arm is called P and the long arm is called Q. Short arm is called P and long arm is called Q. Further, if you see, this is labeled as sister chromatid, and in the end you can see there is telomerase written. So telomeres. So what are telomeres? Telomeres are basically the end of chromosome. Telomeres are end of chromosome. Here, remember, telomere protect the chromosome by the end, right? Telomere is just like the protection. It will protect the chromosome. Protect the chromosome. Telomere will protect the chromosome. Now, this basic part of chromosome we have seen. Now, before we go further into the detail of some point, let's first, I will write all the keyword which we have learned here so that it remain in your mind. very clearly. So first thing what we have seen, we are talking about the parts of chromosome. So one of the important part of the chromosome was centromere, right? Centromere. Now, just think, where is centromere present? So, just see quickly. This is centromere, right? This is centromere. Fine. Then, centromere is also called as primary constriction. Primary constriction. Then, second is arm of chromosome. Arm of chromosome. So again, if a chromosome has 2 chromatid, how many arm? 4 arm. If it has 1 chromatid, then 2 arm, right? See, 4 arm, 2 arm. Then chromosome can have 1 chromatid or it can have 2 chromatid. When it is 2 chromatid, means it is in which form? It is dividing. And when it has 1 chromatid, then it is non-dividing. Non-dividing, right? Then the end of the chromosome has telomeres, right? It is telomeres. So telomeres, what it does? It protects, right? Now 2 chromatid of the same chromosome, 2 chromatid of the same chromosome are called sister chromatid. Sister chromatid. So this point is clear to all of you or not? Labeling point is clear or not? If it is clear, then I am going to ask one question, right? This question will be with the help of diagram. Last class also I have drawn one diagram. So just quickly once go through this. Quickly go through this. I am going to draw a diagram and you have to label it properly. Very good. So let's start. Here if I say now here is the chromosome, right? Now I am writing, I am labeling it. You have to just mention what is A, what is B, what is C, what is D. So let's start from here. What is A? You have to just write A and answer. Then what is B? Then what is C? What is C? Tell me, what is A? A is centromere. Very good. Rao has given answer. Lashya, Chandrika, Venkata. Very good. A is centromere. Very good. Now, I am asking B. Yes, B. So now, B is the arm of the, yes, Pinky is telling B is long arm. Fine. Arm. See, here if we say B, so B you can say arm of the chromosome, or you can also call them sister chromatids, right? Sister chromatids. Now end of the chromosome, as I said, end of the chromosome has what? End of the chromosome. End of the chromosome is, see, one thing is that you can say this as the long arm, this as the short arm. Yes, that is also right. That is also right. Of course, it is a long arm, it is the short arm. We can definitely write it. But with reference to the end of chromosome. Yes, very good. Shakuntala is correct. Chandrika is correct. Very good. So it is telomere. Telomere. And this is the centromere. Remember, in your question, many times NEET try to confuse you by writing, instead of centromere, they write centrosome. Now, centrosome is totally different, right? Centrosome is made up of microtubule. Centromere is the part of chromosome. Centrosome is not a part of chromosome. So always be alert. Many times they give you questions so that you get confused, right? So now, if this part is clear, then let's move ahead and see further some more part. Here one more thing I will like to mention, but here let's see. Now here, can you all understood this point? We have seen sister chromatid. This is the centromere. Now, if I am drawing here a centromere, just see, I am drawing a magnified diagram of centromere. Now, each centromere has this two structure which is called, this is called kinetochore. So, if a chromosome has two chromatids, so how many kinetochore? Two kinetochore. If it has one chromatid, then how many kinetochore? Only one. So number of kinetochore is equal to number of chromatid. In general, if I say each centromere has two kinetochore, so that it is correct. But if I say this diagram, here how many kinetochore? So you will say here only one kinetochore. Only one kinetochore. Here how many kinetochore? So here you will say two kinetochore. Two chromatid, two kinetochore. But in general, in terms of a statement, if I say, so you will say each centromere has two kinetochore. Now, what is the role of this kinetochore? So this kinetochore will go and bind to the microtubule during cell division. They bind to the microtubule during cell division. They bind to the microtubule during the cell. See, here this is the one centromere, right? Inside is two dense structure called kinetochore, and from here we can see that several ray-like structure is coming out, and that ray-like structure is called microtubule. So if I ask what will combine with the kinetochore, so here the diagram, see, these are what? These are microtubules. Microtubules will go and combine with the kinetochore. Is this point clear? So each centromere has two kinetochore. So now we have understood it. Now we have also seen that here almost this arm and this arm is equal, right? So now many chromosome can have centromere at the center, exactly at the center. In some it will be little away from the center. In some case it will be toward the end. Some it will be at the end. So chromosome is divided into different types on the basis of position of centromere. And here it is. Now see here, tell me, in which case, let's say I will put it A, B, C, D. Now tell me in which case centromere is at the center. In which case centromere is at the center. At the center. Let me see who answer first. Centromere at the center in A, B, C or D? Lashya telling A, Preetham A, Srinivas A, Chandrika, Nikhil, Tanish, very good, Pinky, all of you are telling A. Of course, it is clearly visible that it is present in the center, although some of you might feel that this is a little shorter and this is a little longer, right? So thus that is just a drawing confusion, but it is at the center. So the cell, the chromosome in which the centromere is at the center is called metacentric, right? So here I will write in metacentric at the center. At the center. At the center is metacentric, right? Then second one, if I say submetacentric, so it will, what we will say here, it is near the center. It is near the center. Near the center is submetacentric, right? Submetacentric is near the center, whereas acrocentric, it is near the end. Near the end. And telocentric at the end. At the end. So understood these four types of chromosome. Chromosome can be metacentric at the center, submetacentric near the center, acrocentric near the end, and at the end is telocentric. So these are four types. Now how NEET ask a question? NEET will give you this diagram and then they will ask you whether it is submetacentric, acrocentric, or they will give you all the four diagram and you have to identify whether it is submetacentric, acrocentric, etc. Is it clear? Now, so this is the four types of chromosome. Is this point clear? So from here, if question is asked, you all can easily answer. So now, see, this is again one chromosome. So one more part is left here, and that is called satellite. Not in all the chromosome, but in very few number of chromosome, an additional structure is present which is called satellite. And satellite is formed by the secondary constriction. See, the centromere we call as the primary constriction, and satellite is formed by the secondary constriction. So remember this point. Secondary constriction is not present in all chromosomes. Not present in all chromosomes. Same goes with satellite. Satellite is also not present in all the chromosomes, but centromere, it is present in all chromosomes. Primary constriction, also the centromere. Then you see telomere, we have just seen that telomere is the end of the chromosome. So this point is clear. So this satellite has certain character. It is non-staining structure. It is non-staining structure, and it is present in few chromosomes, not all chromosomes. It is a non-staining structure which is present in few chromosomes. Later on we will study, in second year you will study that this satellite will be helpful in DNA fingerprinting. We will see that in the second year, that how it helps in the DNA fingerprinting. So now everything is clear. So lastly, let's quickly do some question. Although the time is up, so I will be very quick and fast. So just 2-3 questions we will do and we will stop. So all of you are ready for the question? Let's do some question. Now here, which structure is involved in the synthesis of ribosomal RNA? Just we have seen rRNA. Quickly answer. rRNA formation is the function of rRNA is the function of, yes, very good Chandrika. Venkata, very good, very good. So now all of you are answering B. Yes, it is the nucleolus which is called, which helps in the formation of rRNA, and that's why it is also called ribosome factory. Ribosome factory. Then next question, what is the primary function of nucleus in eukaryotic cell? Function of nucleus. What is the function of nucleus? Function of nucleus. Let me see who will answer first. Yes, Shankar has given first correct answer. Yes. Shankar has given correct answer. Shakuntala, correct. Yashwant, very good. Right. So C is the right answer. Storage of genetic information. Inside the nucleus, what is present? DNA and chromosome. So nucleus has very important role in the storage of genetic information. Next question. Point where two, it is from the structure of chromosome. First here, point where two sister chromatid are joint. Two sister chromatid are joint is called, yes, Preetham, very good. Preetham is giving right answer. Ritika, Tanish, very good. Pinky, correct. Srinivas, correct. Ganpati, correct, very good. So it is, see, the point where two sister chromatid meet. This is the two sister chromatid. So they meet at this point, and this point is called centromere. Telomeres are found at, where are they found? Telomeres are found at the center of chromosome, end of chromosome, side of chromosome, or inside nucleus. Correct answer will be, very good, Tanish, Srinivas, Faz, Preetham, Misbah, very good. So it is the end of chromosome, right? If this is the chromosome, so the end will be telomere. The structure that protect the end of chromosome is, again, same answer, right? The structure which protect the chromosome is, yes, again the answer, telomere. Telomere, basically it protects the chromosome. Its function is protection, right? Then chromatin in the nucleus is made up of, chromatin is made up of, quickly, chromatin is what kind of a structure? Nucleoprotein. So it is made up of protein and nucleic acid. So it is made up of protein and DNA. So A will be the right answer here, right? Next question. Which of the following is not a function of nucleus? Not a function of nucleus. Storing genetic material, it is the function. Controlling cell ability, yes. Synthesizing protein. Synthesizing protein is the function of ribosome, not nucleus, right? So this one is the right answer. During cell division, chromatin condense to form. Chromatin condense to form. Chromatin form, just we have seen during cell division, chromatin form. Very good, very good. So chromatin form chromosome. During the cell division, chromatin form chromosome. Right? Chromatin form chromosome. So these all questions are from the nucleus. Which of the following structure is found inside nucleus and involved in the production of ribosome? Again, ribosome means I am asking about ribosome factory. Ribosome. What is ribosome factory? So, nucleolus is the ribosome factory. Very good. Last question. Nucleus is surrounded by double membrane structure called, very simple, nuclear envelope, which is made up of outer, inner and perinuclear space. Now, so till this point, this is related to nucleus. I also had prepared question for NEET. As you can see here, these are the questions from NEET 2024 from cell chapter. So this we will do in the next class, right? So till then, you go back, you revise it properly so that you can answer all these questions easily. So we will do it in the next class. Till then, keep on reading NCERT, keep on solving question, revise, and if needed, see these videos again and again to prepare notes. Thank you all of you. Have a great day.